Hello everyone and welcome to this channel. In this video we will talk about pharmacodynamics and how drugs show their effect on our body. So in contrast to pharmacokinetics, we can describe pharmacodynamics as the actions of a drug on our body and how the drug concentration can affect the magnitude of the response. So basically, drugs shows their effect by interacting with receptors. So in pharmacology, receptor is any biological molecule to which a drug binds and produces a measurable response. And these receptors, they can be enzymes, nucleic acid, but most commonly they are proteins. And these receptors exist in at least two states, which is the active form and the inactive form. And these states, they are in reversible equilibrium with each another. And when an agonist binds to the receptor, it causes the receptor to shift from the inactive form to the active one, so that it can produce a biological effect. And how much this biological effect will be, it will directly depend on the fraction of the activated receptor. These receptors are mainly divided into four families. The first one is the ligand-gated ion channels. So the channel is usually closed until the receptor is activated by an agonist, which causes to open the channel briefly for a few milliseconds. In this type of receptor, it is the extracellular portion of the receptor that contains the ligand bind inside. So only hydrophilic ligands can interact with these receptors. The next type of receptors is the transmembrane G-protein coupled receptor. In this type, the extracellular domain of the receptor contains the ligand binding area, and the intercellular domain interacts with a G protein when it is activated, or it can interact with another effector molecule. And they usually contain there are three types of G proteins, which is the GS, GI, and GQ. And all of them, they contain three protein subunits, which is the alpha, beta, and gamma. When it is not activated, the alpha subunit is bound with GDP. But when the ligand binds to the receptor, and they separate between them, and the alpha subunit binds to GTP, while beta and gamma dissociate separately. And these two complexes can interact with other cellular effectors like enzyme or protein, or it may be even ion channel, and they will be responsible for further actions within the cell. An example of this effector is adenyl cyclase, which is activated by GS and inhibited by GI. And when it is activated, it produces the second messenger cyclic adenosine monophosphate. On the other hand, GQ can activate phospholipase G enzyme and produce two other second messengers, which are inositol triphosphate and diacylglycerol. And these second messengers can amplify the effect. The third type of the receptor is the enzyme-linked receptors. And this family of receptors, they consist of a protein that may form dimers. And these receptors, they possess the tyrosine kinase activity. And when these receptors are activated by a ligand, they can phosphorylate tyrosine residues on itself, and they can also phosphorylate other proteins. And this will again result in multiplication of the initial signal. An example of this type of a drug that binds to this type of receptor is insulin. The last type is the intercellular receptors, and these ones, the receptor is entirely intercellular. So the ligand must diffuse into the cell to interact with the receptor, and for it to move across the target cell membrane, it must have sufficient lipid solubility. And their main target are the transcription factors inside the cell nucleus. Thank you for watching. Please don't forget to subscribe.